All right, we've begun. We've only just begun to talk. You didn't get that right. Hey, in the back. Hello. Good morning. How are you? Nice chatting with you. We don't have a, we let's just get through this as much as we can. Uh, we're finishing up this, which is smooth muscle. There's not a lot left to say about it. I guess the biggest single difference is one, their singles is redundant. Biggest difference is principally it utilizes extracellular calcium. It doesn't mean there's not intracellular calcium, but the big player is extracellular. It's a big distinction. There are no neuromuscular junctions. There's no motor end plate. They call them varicosities. That's where the nerves contact this area and have a bulbous enlargement, which is by definition what a varicosity is, even though we classically see it in veins. And it's a synapse, except because it's wide, they call it a diffuse junction. And when we get to autonomic, you'll see the principal neurotransmitters are either acetylcholine or norepinephrine. When we get to the neurotransmitters, because those are the principal, those are the ones, for lack of a better way of expressing it for now, as that are secreted by or released by the autonomic nervous system, which we have not gotten to. So that's part of it. They look like this. This is a really good illustration. So these enlargements have synaptic vesicles. The enlargements have mitochondria. It's not a whole lot different. It's just sort of, when you say diffuse junctions, it's just juncture points where it diffuses. And again, it's not nearly as rapid. The distinction is skeletal muscles are very rapid and very strong. Smooth muscles are all about endurance. They aren't necessarily as strong as skeletal muscles, but they effectively go continuously. That's the difference. Smooth muscle does not have the elaborate sarcoplasmic reticulum. They do not have the T tubules. Okay, it's not nearly as well developed. It does store intracellular calcium, but it only has a limited amount. Most of it is all about extracellular calcium. And you're going to see, this is a term you'll see more than once, called caveolae. It's like an indentation or an infolding in the plasma membrane. This is still called a sarcolemma, and it has a lot of calcium channels that open to allow the extracellular calcium in. So this would be a different kind of a channel. This would be more in the line of a ligand or chemically gated channel that allows this influx of extracellular calcium. So they have a pretty good illustration of here, though they are calling it a voltage gated calcium channel. I guess they're indirectly indicating that. That's interesting. I wish they were a little bit more expressive in the diagram, and I hadn't really noticed this about, here they're only showing you what occurs. So I am guessing that where the neurotransmitter acetylcholine or norepinephrine is elsewhere, alters the membrane and creates a voltage gated channel. So we have to go with that, along, and allows the extracellular calcium in. And then at the same time, we have some release within this limited sarcoplasmic reticulum space. Okay, the calcium entry from here triggers calcium release from here as well. Okay, so it's a combination, but the vast majority is extracellular calcium. So the muscle fibers are typically communicate with one another because, unlike the isolated nature of skeletal muscles, one group over here, there's arranged in sheets of these individual cells, which are not nearly as large or as complicated, I think, as skeletal muscle. So gap junctions sending signals from one another. Okay. The skeletal muscle fibers are isolated. The gap junctions basically allow that depolarization to spread. And so you cannot see striations. That's where, even though there's overlap, the thin to thick overlap is something along the order of 12 to 13 to 1. Where in skeletal muscle, it's 2 to 1. And because there's so much thin in relation to the thick, you don't see striations. So I frame it as no visible striation, Certainly no sarcomeres. It's a whole different ballgame, as you will see. But it does have thick and thin filaments, so we still have actin and myosin in play. So the big difference is thick filaments are fewer and have myosin heads along the entire length. We don't have that isolated area where the head is located. That's there. Again, 1 to 13 compared to 1 to 2, thick to thin. The thick filaments have heads along the entirety, making the smooth muscle as powerful as skeletal muscle. So it doesn't indicate, it's not that it's not strong, it's just the speed and the arrangement of it that's there. It does not have troponin. It has tropomyosin, but not troponin. It relies, the whole thing is triggered by the calcium, essentially the vast majority extracellular, to bind to a protein that changes shape, not unlike what troponin does, but arranged differently. It's called calmodulin. So there's certain names to remember. We have calmodulin to remember. We have varicosities to remember. We have caveolae to remember. We have the absence of troponin to remember. Just stuff like that. So here, you're going to see a typical smooth muscle has a nucleus, usually just one. It's tapered at both ends. So that's that characteristic look that it has. Wider in the middle. And you can see the arrangements that are there. The caveolae are these areas where the calcium will contact and get in, and you begin to see these, you can see the channels that are there, you can see the nucleus are there. What they have connecting, these are intermediate filaments that are arranged in this somewhat, the way I would describe it in the book does too, it's a lattice work. So it's like you have on your house or maybe around, older homes have a lot of lattice work. Those boards that kind of overlap each other and that type of stuff. The older homes have a lot of those things. And they're connected where they crisscross with these things called dense bodies. They are the analogous structure to the Z discs that you will see. So they're arranged, so the thick and thin filaments that you're seeing are arranged spirally. So the muscle contracts in a corkscrew manner. Twists like that. The intermediate filaments and dense bodies, so they describe it as I would lattice-like. They're non-contractile, but they give it effectively in its shape. The dense bodies anchor them, analogous to or corresponding to the Z discs. So the sarcolemma between Z bodies, between dense and bodies bulge outward. So it does this. It kind of widens in the middle. And so depending on the arrangement, the way the sheets are, again, when they're concentric around the lumen, they squeeze, and when they're longitudinal still, they push. So it's squeeze, push. I mean, my favorite analogy, the toothpaste at the very end where you got to squeeze it and you fold it, and squeeze it and fold it. That deal. Love that. I was going to make a bad joke, so I will not. No, it had to do with toothpaste. It's all. Not, I'm trying to think of a clever pun with toothpaste involving something like Crest or, that's it. You saw that illustration before. So, and they're just showing you the distinctions that are there. So actin and myosin filaments are there throughout. They have those caveolae, those indentations. I mean, there's really, they have endomysium, but that's just about it. Cardiac we'll deal with later. So smooth, equivalent to cardiac muscle, but much less voluminous. I mean, you're talking maybe at best less than 10% of its volume. Some sarcoplasmic reticulum contacts the sarcolemma. Okay, it's unitary. So skeletal does not have gap junctions. Cardiac certainly does, because that's how that signal is conducted, and we get to the physiology. And it's a unitary muscle, but the gap junction signals other muscles. Okay, so it functions as a multi-unit, and it's involuntary. And you're going to see we have, and a lot of them, these when they talk about pacemakers, a lot of these work with stretch receptors. They respond to materials going through, and it sets up a sequence, almost like a chain of events that occurs. Local chemicals, hormones, stretch receptors. You're going to see acid in the digestive, pH controlled, osmolarity level controlled, stretch receptor control. There's a lot of different controls that work, particularly gastrointestinal. Calcium from the extracellular fluid. Here's the calmodulin, and that's going to change shape, okay? And you can see how relatively slow, much slower than cardiac. And again, to me, it's all about endurance, where cardiac is the blend between endurance and strength, okay? And yeah, they're mainly aerobic. Slow, synchronized contractions, coupled by gap junctions. The action potentials are transmitted. Again, pacemakers, when when an area is stimulated, it triggers the rest. Okay, and there's a variety of things that do that. And again, so a lot of times that's where a lot of the medications, when you're someone's stomach is overactive and you're dealing with diarrhea, think of that where maybe Imodium would work. Let's have something available over the counter. So contraction is similar. The actin-myosin sliding filament. The trigger is intracellular calcium. We need ATP. Contraction stops when the calcium is no longer there. Mostly extracellular. Here's what occurs. The sequence is important. I know that I did send you a study guide. Aren't you lucky? I'm looking like the 23rd or the 25th, that neck of the woods, because I try to avoid Fridays because these sport people have things to do. So start marking that down. We'll know a little bit better as time goes on. And the calcium binds to the calmodulin, not the troponin. The calmodulin then, its sequence activates what are called myosin kinases. And those kinases, okay, are the ones that activate by phosphorylation the myosin heads and effectively crossbreed. So the intermediary is a little bit different. Calcium and calmodulin rather than just calcium on troponin, and then this intermediary of myosin kinase, which is activated. So relaxation, the calcium has to detach, be actively transported out, as well as back into the sarcoplasmic reticulum, and take away the phosphate and deactivate the myosin. You've already seen enough of this in cell and molecular related courses. What do we do with ATP when we break it apart? We take that phosphate, that high energy phosphate, and that's usually that's what kinases are about. And phosphorylases take it away, right? See, same stuff. But I'm not going to ask you that. Thankfully, that's a different course. And they just show it to you here. Calcium ions. Again, voltage gated or non-voltage gated calcium channels. I'm not going to ask you about them because when you have both answers that are there. There's the inactive calmodulin. It changes shape to activate it. Then you can see it activates just by their diagrammatically. It's activating the kinase, and the kinase is liberating the phosphate to do it on the myosin heads, and voila, there we go. So slower to contract and relax, that's typical. Start slow, end slowly. Maintains it for a long period of time, endurance. Not a lot of energy, slower ATPases. Myofilaments can alter themselves to save energy. Okay, and there's just like there's tendon when we talked about tone in muscle tendon complexes, muscle tone, we have a smooth muscle tone. There's always tone in these things. In blood vessels, that's the lower number on your blood pressure. The tone is the resting tension in a blood vessel, that bottom number. And that's the most critical number for high blood pressure. Nothing is even close. Such an important measure. So when we look for I mean, clinically, you look for those measures. If somebody's over 100, oh boy, they got big time hypertension, high blood pressure. And I mean, if it's somebody, all of a sudden you see it is barely, you can barely measure it, or you can't measure it, then you're starting to tend toward shock. So it's very significant. You'll see down the road, it is controlled by the sympathetic nervous system. That is a highly significant feature. Okay, very important. So there's typically some degree of contraction. That's blood vessels. The same thing digestively. It doesn't conditions where it loses its tone, then stuff doesn't move, and more or less it's moving all the time. A lot when you're at rest, considerably less when you're active. If you're very active, it may stop for a brief period, but usually those events are relatively short. So again, we'll get into it. Neurotransmitter increases the calcium concentration, depends on the neurotransmitter. And this is the idea. When we start looking at this, and we'll get into this with its controls by the autonomic nervous system, it depends. Sometimes they're excitatory, sometimes they're inhibitory. They'll either make it contract or they'll make it relax. More likely or less likely. So the nuances, I love teaching autonomic nervous system. I think it's it's where most of, if you know somebody who's on high blood pressure medicine or heart medicine, typically asthma medicine, you're going to see it there. Asthma medicine deals with smooth muscle in the respiratory passageways. And everybody here knows somebody who has an inhaler. They just do. So, again, some of them in response to chemicals will play a role. And that's what I mean. It can be hormonal chemicals. It can be in response, think respiratory. In response to high levels of carbon dioxide, that will alter the pH because it alters our buffering system, and low oxygen. You're going to see that trio again and again. And order is important. Oxygen is really important. We can't live without it. Typically, it's not the thing that triggers. It's typically carbon dioxide and pH play a more significant role. What makes you want to breathe in is not low oxygen, it's high carbon dioxide retention, which in turn alters pH, particularly in the central nervous system. That's what happens. If you have to rely on low oxygen to make you breathe, you're probably not going to make it. That whole oxygen utilization curve, that dissociation curve, not good. Some smooth muscles respond to both neural and chemical stimuli. I'll buy that. Stress related, stretch them. So those are the stretch receptors. Length tension changes. You can contract between half and twice its length. So organs have huge volume changes without becoming flabby when relaxed. You'll see when you get to the stomach, maybe you remember this for those who were there for the anatomy portion of the program. Your stomach empty is about less than 2 ounces. Filled, it can be a gallon. So it can stretch and still go back when it finally empties. Okay? So you're doing a great job. I tell my wife, you got to give credit to the design folks. Your design team is working pretty good. By the way, make it 3-0 as the final 2 numbers. The 3 for Seattle and the 0 for. I could win a boatload of money. Pool, Super Bowl, you've heard of it? You ever get one of those with 100 square grids? And sorry. Don't get that gambling thing could be bad for you. Yes, my son and my oldest and my son-in-law must be doing pretty well because they fought over a check at an expensive dinner. I said, shut up, Dad. I know when to take yes for an answer. So I don't get crazy about the fiber arrangements and stuff like that. Don't care. Don't care. Don't care. Oh, thank goodness we're done with that. I love ending units. I love starting new units. So here comes a really important one. I have it loaded. This is 11. This is nervous system. Oh boy. Oh, and boy. Is there a lot here? So we spent a fair amount of time in anatomy for the folks who were here on the anatomy of the nervous system. We didn't deal so much with cellular anatomy. We're going to look more deeply at the cell of the nervous system, the neuron. It's not the only cell. The nervous system is unique in the sense that all the other systems utilize the same connective tissue. They have collagen and elastin and reticulin, dense regular, dense irregular, loose areolar, all that stuff we learned about in tissues. You have a whole new cadre of cells and players in their arrangements when we get to the nervous system. The nervous system have their own set of connective tissue cells that make the connective tissue that surrounds, whether it's the central or peripheral nervous system. As you will see, they're called glial, G-L-I-A-L. They're coming up right away. G-L-I-A-L, which means collectively call them neuroglia, nerve glue, or collectively glue. When you hear about someone with a nerve tumor, oh my God, so-and-so has a brain tumor, it's terrible. And it is a terrible diagnosis. It's not the nerve cells that have the tumor. Remember, they're like muscle cells. They're a cell that has exited the cell cycle. It's a cell that, when it's damaged, is going to be damaged and repaired by scar tissue. It's only in cells that can still replicate that we see cancers evolve, other than the rarest of rare cases, maybe developmentally, that would be a different. So you're going to hear the common ones that you hear the most of are called gliomas. Collectively, it's involved in that connect astrocytomas. You'll see these as they come up. So it's the master, and we talk about this. We have two major control systems. The nervous system, rapid changes. The endocrine system, not as fast. By no means slow. So in the nervous system, fractions of a second, microseconds, milliseconds. Endocrine system, seconds, minutes, days, hours, even longer. It varies. So electrical and chemical signals, basically it's an electrical system. When the nerve communicates with another nerve, it's chemicals. You've already learned about some of that. They're neurotransmitters. The one that they're the most of, acetylcholine. And we've already got that one in our head. We have it in our toolbox already. So very, very rapid, very specific, very often immediate responses. Very straightforward. Simplest circuit, at the minimum, has 3 nerves: a sensory nerve, an integrating or associated nerve, and an effector. So you have a sensor to bring in the information, we call the afferent or incoming portion, something to integrate it or associate it. There could be a bunch more of those. And an effector, something that's going to have an effect in a motor sense on muscles or glands to produce a response. That's the simplest circuit. Even the knee jerk. Because again, when you're sitting there like that and boom, they test your reflex, that was a sensor. The motor was the response. But you know if somebody's whacking your knee. So there's some information from that association nerve to say, oh, somebody's testing my reflexes. I felt that in my knee. And if you didn't feel that, you would normally feel that unless there was some other issue that was going on. Think of it that way. So here it is. I see that. I'm thirsty. Well, in here, the fact that you were reaching and getting a drink, you're aware of that. So in the old days, you think with sensory, like a knee jerk would just be pure sensory and motor. That's okay. And we think that we have so many sensors and so many effectors. Not the case. 98% of our nerves are association nerves. It's all about, they're the information processors. How we process it and how we respond to it is the critical piece. All the decision making that goes on. The reflex is just the simplest circuit that we have. Think of all the things that are involved in what you're doing now. Okay, so hopefully listening to me and processing some of the information. I know it's Friday. I'm not naive. I know you have you're going out. Where are you watching the Super Bowl? Here? Quiet night at home, huh? You got to get out more, dear. It's all right. I'm the ultimate homebody. My wife will look at you. Do we have to go out? It's miserable weather. It's cold. No, we don't have to go out. The only one of us has to go out is the dog, and he just has to go out, as you know. So the nervous system is divided into, we had this central, this is anatomy, brain and spinal cord, period, end of the discussion, definition of the central nervous system, integration and control, sure, peripheral, anything that protrudes from the central nervous system. Nerves, and basically it's the starting point, cranial nerves and spinal nerves. We're not going to go over all of them again. That's anatomy. I know for a couple of you are taking this out of sequence. That's part of the stuff you have to review. I am more than happy to have questions. You can have, increasingly more these days, students are less eager to ask questions in class. But you know me, I respond to emails right away. If you didn't know that, I do. I got nothing better to do. Okay. I don't tinker in the house. It's not, oh, I've got a carpentry project. Are you kidding me? It's the simplest thing to do. I couldn't even figure out how to put a new filter into the furnace until the guy came yesterday to clean it. Show me. He goes, I just pull it apart. Really? Why didn't I know that? My wife usually has a way of expressing that to me. Why I was so surprised. She said, you're so dumb sometimes. It's okay. I'm used to it. So we've seen that diagram before. Sensory or afferent. It's coming from the body. Okay, skin, muscles, joints. Somatic, visceral means it's coming from organs. We have a lot less specificity there. Motor, transmitted, muscles and glands. If it's somatic, if it's going to organs, we call it autonomic or automatic if that works for you. That's fine. I use autonomic because that's how it's traditionally represented. Somatic nerves conduct impulses from the central nervous system to skeletal muscle. They're voluntary in nature. But now we're at the point, even though we've discussed how those works, the autonomic just we looked at smooth, cardiac, glands, really important. And that's what we'll look at, the divisions that are there. These two divisions that we call it dynamic antagonism. They work opposite, but they work together. It's a lot of that. Oh, push a little harder. Up. That's enough. Pull a little bit more up. Not too much. And that's the beauty of art. That keeps us. They're all about homeostasis. Wonderful system. You've seen that diagram. We'll get there. Glial cells. So the glue that holds it together. At least I want to introduce it. There are 6. Bless you. 6 of these cells. Classically, 4 of them are associated with the CNS. 2 have similar, one has very, very similar, one we're not necessarily clear of, in the PNS or peripheral nervous system. So we have astrocytes, by far and away the most abundant, the most multifaceted in their role. And we think, and I emphasize that, we don't know, we're called satellite cells. We have oligodendrocytes. You're going to see down the road when we get to it, if not today, back Monday, they're all about myelin. Same thing is true of what's called the Schwann cell in the peripheral nervous system. Two ends. Has nothing to do with the guy with the frozen foot. So that's okay. About myelin. The other ones really don't have an equivalent over here. We have microglia, very tiny ones, and we have, it'll get to me. Ependymal cells. God, I thought I was losing it. Again, my son who just called me is driving back, wants my views on the Super Bowl for his wagering. I should give you his email in the military email in case I lose it. He'll want to know. He's ready to put me in the home soon. My dad walking around undressed. Does he know what he's doing? Enemies. So if you get a military email from an Adam Stein, he's looking for information. It's true. So glial cells wrap around delicate neurons. Nerves themselves we'll get to. Here are the categories. Astrocytes, as you will see, and we'll get to what we can to today in introducing it. Microglia, ependymal, and oligodendrocytes. See, I didn't even have to look at that to remember. The astrocytes are really a big player. So they do so much. Okay, they cling to nerves, they cling around in the area of synapses, they cling around blood vessels. They are part of this complex that creates this pivotal thing we've discussed called the blood-brain barrier. Capillaries are unique in the central nervous system because they have an abundance of tight junctions. So only materials and a very, very limited amount of ones that are water soluble can get through. Very, very limited. Other than that, they have to be membrane small and have an abundance of non-polar bonds. So oxygen, carbon dioxide, medications have to be. There's a whole subset of medications that can go through the blood-brain barrier, and it's because the astrocytes are the pivotal part of it, in addition to the main part is certainly tight junctions of the capillaries. After that, you'll see what the astrocytes do. So they're supportive in nature. The role of exchange you will see demonstrated more graphically. They play a role in development. They play a role in whatever is allowed to encounter nerve cells. So they play a role in limiting things that can go there. They can be altered by nerve impulses and neurotransmitters. They're influential in their function, and they even play a role because of all of this in information processing. They are big players, and there's more of them than any other. Astrocyte, because it has star-shaped processes, cyte for cell. Look, here's a capillary. They surround it. Here's the nerve. They surround, they're in country. So elements will have to come through the capillary into the astrocyte, along the astrocyte until it's delivered effectively to the nerve cell. That capillary, again, very, very limited as to things that can get through that boundary. Small nonpolar molecules for sure. Medications that are, and that's what in pharmacology, you have to do those medications. Unfortunately, a too common diagnosis, Parkinson's, we know it's related to an absence of dopamine. But we can't give people dopamine. Dopamine is a water-soluble, larger molecule. We give a precursor traditionally called L-DOPA. There's other medications. The L-DOPA is compatible and will cross that blood-brain barrier. if I didn't mention it to you, there's a movie from circa 1990 with the late Robin Williams and Robert De Niro called Awakenings. It's not an all-time great movie, but it talks about a physician who was using that increasing. It's a good watch, so to speak. It's sort of interesting. De Niro does a really good job. I'm not necessarily his biggest fan as an actor. Uh, Robin Williams is brilliant as he always was in these roles. And the lady who's the voice of Marge Simpson does this role. She's in that as the nurse. She's got that sort of nasally New York kind of a vibe that I could do if I wanted to do, but I don't. But I could. So don't underestimate microglia. They're sort of scavenger cells that are kind of like play the role of white blood cells, white blood cells that are there. When there's damage, they can phagocytize, they can scoop up debris. So they have an important role, and this is what they mean. This is what have you, these like thorny processes that are abundant. It's very white blood cell. Like white blood cells have a lot of appendages that can sample environments. If you go back and look, when we talked about an anatomy and we looked at dendritic cells, dendrites are appendages that are classically about nerves, but we have some in the skin, we have them in the liver. Okay, they're cells that are samplers, and this is more of a defensive nature that's there. Ependymal cells vary somewhat in shape, may or may not have cilia to circulate cerebrospinal fluid. We see those when we did the brain dissection, the sheep brain dissection. I pointed out to you some areas when we saw dark material. That's really coming from where we see this sort of confluence in external circulation covered by these cells, and they play a role in the generation of the cerebrospinal fluid. So between cerebrospinal fluid in those cavities and bathing the central nervous system cells that are there. So they cover these areas, if you go back and look, that are there. And the cilia are there, and basically they create almost a circulatory environment over them. And then the oligodendrocytes, they're the myelin sheath. There's a significant difference in myelination of white matter in the central nervous system and white matter in the peripheral nervous system. You get the microglia, what they cover, they have other things sort of like they cover little protrusions of the arachnoid villi that are present, that play a role. There's a number of these areas, and we see them, if you remember, particularly so just before I get into the oligodendrocytes, particularly in the ventricle areas. There's a lot of protrusions where not the microglia, where I beg your pardon, they should be in the appendix. We have in the peripheral nervous system, you have a lot of room. In the brain, you do not. So what you're going to see that the myelin sheaths made by the oligodendrocytes, they're very similar, and then they have the gaps that are so critical, and then I might have mentioned to you before, I'll mention again. But up to about 60 nerves can be engulfed by oligodendrocytes, different oligodendrocytes at different spaces. This is what I mean. This is just looking at 3 nerves. There's typically more. Here's the oligodendrocytes. It's myelinate. Basically, its cellular membrane can extend and engulf up to 60 nerves. So you can pack axons from 60 different nerves together. We don't have a lot of room. The Schwann cell in the peripheral nervous system surrounds just one axon. Okay? But very, very similar roles. That cell membrane effectively makes up what myelin. Myelin is nothing more than a fatty layer with embedded proteins, very much like a cell membrane. The satellite cells are they surround cell bodies in the peripheral nervous system. Remember, we don't use the term nuclei for nerves. We call them cell bodies. I'll go back. And I'm sure you're going to see that as we look at the nerves themselves. Function similar to astrocytes. When I first started teaching this, 25 years ago, with anatomy and physiology, more than 25 years ago, the function was unknown. So, this is now more current theory. Schwann cells, also known as neurolemmacytes, the neurolemma was the classic term for the sheath around the nerves, surround peripheral nerves and form the myelin sheath there, for sure. Their function is similar to, but their structure is different than oligodendrocytes. And they really are pivotal in nerve regeneration. We don't see that in the brain. We see that in nerves here. We didn't understand that nerves could regenerate until about maybe 30 to 40 years ago, when those of us in medicine and those of us who did surgery began to hear about things called growth factors. And nerve growth factor was something that would emanate from cut ends of nerves, and they would start to grow back together. You might recall me telling you this: when we would operate an area, you can't help but cut axons. You can't. And I have people routinely, I can't feel anything. And you try to reassure them. And you have to tell them beforehand that you could lose sensation. And then I have people who come back 6 months, a year later, oh my God, I can feel it again. It doesn't happen with everybody, but it's part of what goes on. That's part of the nerve growth factor. Nerves have an unusual tendency that can somewhat be problematic of seeking each other out peripherally. It's why people will have things called stump neuromas when we have to do amputation. And a lot of times we'd have irregular sensation. It has to do a lot with scar tissue. And if you ask anybody who does surgery, the one thing you can't plan on is how scar tissue forms. I would do bilateral identical procedures, and one would be perfect, and the other one would be, oh my God, I can't feel it. And there'd be abundance of scar tissue. It's just the way it is. And you must be, if you're going to go into that field, in a medical field, be very aware. And I was very much, that's why I didn't make a lot of money. I would tend to talk people out of it. I didn't believe in trying to convince people that they had to have operations. It's okay. I have to look at that mirror and look at and like the person I'm. And by and large, I do. But it didn't necessarily make me a ton of dollars. But really, the peripheral nerves, and we'll see evidence of this, and I'll talk about that with nerve growth factor as we think about some of the physiology behind it. But here's the satellite cell, and you can see the difference. The individual Schwann cells and their nucleus surround at these intervals the nerves to create the myelin. And if we go back and look at the oligodendrocyte, the cell body of the nerve is not part of that. It's separate and distinct. It just has an enormous cell membrane that can create multiple myelin sheaths. Do you remember we talked about these gaps? Did we not? Yes. Do I talk about nodes of Ranvier, which I love to talk about rather than saying Ranvier? It's sort of like I live in North Versailles, I live in North Versailles, where I did cashiering at Target. Oh, you worked at Target? No, I worked at Target. It's okay. Two major neuroglia seen in the peripheral nervous system: satellite cells and Schwann cells. You see them there. The nerves themselves are a little bit different. We will start with this on Monday. So lab in Monday is going to be, I'm going to use some of the technology. I have to talk about the lab, but we need to start getting into eye and ear physiology, and I tend to do the cardiac stuff in the lab as well. Yeah. I've got to divide it, okay, in that regard. So see you all then. Have a great weekend. I don't have a favorite per se in the Super Bowl, so it's kind of nice to watch it that way.